Belgian hospitals, Ziekenhuis Oost-Limburg en the Jessa Hospital. First I will tell some background about my study. Lung cancer is actually the most commonly diagnosed cancer in men and the second leading cause of cancer death in men worldwide. Among females, it is the fourth most commonly diagnosed cancer and the second leading cause of cancer death worldwide. The 5-year survival rate of lung cancer is very poor, namely approximately 15%. And as you all can see, the incidence rate of lung cancer is approximately equal to the mortality rate. As you can see, in 2008, there were approximately 1 million new lung cancer cases in males, and also in 2008, approximately 1 million males died from lung cancer. Then in Belgium, lung cancer is the second most common cancer in men and the third most common cancer in women. The main cause, of course, of lung cancer is the smoking of cigarettes. When a person who has never smoked has very little risk to develop lung cancer. This risk is not completely zero, but it increases with increasing age. This phenomenon you can also see for active cigarette smokers. At an age of 65, the risk is approximately 5% to develop lung cancer, and this risk increases to approximately 15% when an active smoker has an age of 75. This figure also clearly shows that when you quit smoking, this has an important influence on the risk to develop lung cancer. For example, a person who stopped at the age of 50 has a risk of approximately 5% to develop lung cancer at the age of 75. This is much less than the risk that an active smoker has. Also, other causes lead to the development of lung cancer, such as occupational exposure to asbestos or to combustion fumes, and also environmental exposure plays an important role, such as exposure to latent decay products, passive smoking, and to air pollution. The symptoms of lung cancer include persistent coughing, chest pain, dyspnea, loss of appetite, and also weight loss. But as you can see, these symptoms are very general. They are not specific for lung cancer, and they are unfortunately not present in an early stage of disease. Therefore, lung cancer is often only diagnosed in a metastatic stage, but when it's actually too late. On the figure you can see a person who has a primary tumor in the left lung. And he has also a metastatic tumor in the right lung and distant lymph node metastasis. Lung cancer can also spread to secondary organs. Most commonly, lung cancer spreads to the brain, the liver, the adrenal gland, or the bone. When a patient has persistent symptoms, an RX or a CT thorax image will be made. Then when a lung lesion is detected, a lung biopsy will be taken to investigate whether the tissue is benign or malignant. When the tissue turns out to be malignant, it can be a non-small cell lung cancer or a small cell lung cancer. In 85% of the cases, it turns out to be a non-small cell lung cancer. This type of lung cancer consists of different histological subtypes, such as an adenocarcinoma, spinocellular carcinoma, a mix of the above two, namely an adenosquamous carcinoma or an undifferentiated large cell carcinoma. Then in 15% of the cases, it turns out to be a small cell lung cancer. This type of lung cancer has a poorer prognosis in comparison with non-small cell lung cancer because it divides more quickly and it spreads more quickly to the secondary organs. Then the staging of lung cancer occurs by the TNM classification, where the T stands for the primary tumor, the N stands for regional lymph nodes invasion, and the M stands for distant metastasis. The T stages can range from T1 to T4, where T1 is a tumor smaller than 3 centimeters in the greatest dimension. T2 is a tumor between 3 and 7 centimeters. T3 is a tumor bigger than 7 centimeters, and T4 is a tumor of any size that invades any of the following organs, the trachea, the great vessels, or the mediastinum. Then the end stage can range from N0 to N3. where N0 is no regional lymph nodes invasion, N1 is when you have lymph nodes invasion in the same lung than where the tumor is located. When you then have lymph nodes in the lung on the lung highest, that is on number 10. Then the N2 stage is when you have lymph nodes on the same side of where the tumor is localized, and when you have lymph nodes in the mediastinum, between the two lungs, or under the carina, that is on number 7. Then an N3 stage is when you have lymph nodes in the other lung than where the tumor is localized. This on the other side of the mediastinum, this on the other side where the highness is located, just on number 10, on the other side of where the tumor is localized. Then the M stage can range from M0 to M1, and M0 is no distant metastasis, and 1A is when you have metastatic tumor in the contralateral lung, and M1B is when you have spreading to secondary organs, such as to the bone. Then for every TNM classification, there is a corresponding stage. For example, a person who has a tumor smaller than 3 centimeters with a T1 tumor, which has lymph nodes in the same lung, this N1, he has a tumor stage of 2A. On the right side figure, you can see the 5-year survival rate of persons with stage 1, stage 2, stage 3, or stage 4 lung tumor. And we can clearly see that a person who has a stage 1 lung tumor has a higher survival rate than a person who has a stage 4 lung tumor. Thus we can see a clear negative link between the stage of the tumor and the prognosis for the patient. Thus an early detection of lung cancer can lead to an improved survival rate. The treatment options then for lung cancer, well, these are dependent on the stage and the histology of the lung tumor, and also on the general condition of the patient. Treatment options can be surgery, radiotherapy, chemotherapy, targeted therapy, or a combination of the previous mentioned therapies. Then metabolomics is actually a newcomer in the life science field, like genomics, transcriptomics, and proteomics target the genes, the messenger RNAs, or the proteins in a cell, an organ, or an organism, while metabolomics targets the metabolites or the small molecules. These small molecules can be fatty acids, amino acids, or sugars, and metabolomics can actually detect metabolic responses to various stimuli, such as, for example, a disease. This figure shows the central dogma of molecular biology. We have the complete set of genes in an organism. This is called genome. Then you have the transcriptome, the complete set of messenger RNAs, proteome, the complete set of proteins, and then eventually you have the metabolome, the complete set of metabolites in an organism. What is now actually a metabolite? Well, when substrate is converted to a product, this end product is actually called a metabolite. And these metabolites are the end products of the cellular metabolism, and therefore they are the closest to the phenotype of the organism. What is now actually a metabolic phenotype? Well, this is a characteristic metabolic profile of the body fluid, which results from the interaction between the host genome and the environmental factors. NMR spectroscopy is a technique that we use. This is actually an analytical procedure which generates the metabolic phenotype of a body fluid. The most commonly used body fluids are plasma, serum, and urine, because these samples can be very easily collected. The advantages of the technique, well, this technique gives information on a wide range of metabolites in a single measurement. It actually provides a metabolic snapshot. This technique also requires minimal sample preparation. The technique is also rapid, which delivers high throughput with a lot of information. The technique also gives detailed information on the molecular structure. The technique is reproducible and non-destructive. We actually use proton NMR spectroscopy. This technique detects actually signals of hydrogen atoms in metabolites. There's actually all hydrogen containing molecules in the sample will give rise to a proton NMR spectrum. You can see an example of a proton NMR spectra beneath. This is actually a proton NMR spectrum of blood plasma. Why do we use proton NMR? Well, this is the most commonly used nucleus because it is abundant in organic molecules. You can also see the structure of lactate. You can see that there is one hydrogen atom bound to a carbon atom, and on the other side you can see 3 hydrogen atoms bound to a carbon atom. This structure gives rise to 2 signals in the proton NMR spectrum. A proton NMR spectrum of blood plasma contains actually low and high molecular weight components. You can see broad band for macromolecules such as lipids and proteins that you can see here. You can see that we have broad bands between beneath the sharp peaks from the small molecules, and therefore we actually use the Garb-Bursell-Magon-Gill sequence to attenuate the signals which result from these macromolecules and to retain the signals from the small molecules to clearly see these signals. And then you get a spectrum like this, and then actually we separate the spectrum in different variables or segments that we are going to analyze. This is the equipment that we use. We use a Varian Innova spectrometer, and we will analyze the metabolic composition of blood samples with this machine. This actually, what do? We collect blood samples from 2 groups, namely from a control group and from a disease group. Then we measure the metabolic composition of these blood samples by means of NMR spectroscopy. Then we get a lot of data. We actually get a data matrix. Then by means of statistical analysis, we are going to investigate whether there is a difference in the metabolic composition of the control group and of the disease group. And then of course we need to investigate which metabolites and which pathways are responsible for the discrimination. We think that proton NMR spectroscopy has the potential to early detect lung cancer. Why do we think this? Well, literature has already shown that unique alterations occur in the metabolic composition of body fluids during disease. This was already shown for various cancers, for example, for breast cancer, prostate cancer, and ovarian cancer. Thus now we actually need to identify which changes in the metabolic phenotype of blood plasma correlate with the presence of lung cancer. Cell metabolism, actually all cells in the body require energy and they take up nutrients. Thus they use energy in the form of ATP, and they take up nutrients such as glucose, amino acids, and fatty acids. The cell metabolism is regulated by growth factors and by downstream signaling pathways. So when a growth factor binds to the receptor on the plasma membrane, then a signaling cascade is activated. Transcription factors will bind to their target genes in the DNA, and cell proliferation is initiated. Then the metabolism of a cancer cell is actually reorganized to increase anabolic reactions which are linked to cell growth and cell proliferation. When a cell is at rest, there is actually low growth factor signaling at the cell membrane. This actually allows the cell to make sufficient energy and sufficient synthesis of macromolecules to maintain the cellular homeostasis of the cell. Then when there is no ligand present, there's no extracellular growth factor, the cell will lose the expression of the nutrient transporters on the cell membrane. Then the cell cannot take up any nutrients, and then the cell will break down their own organelles. This is actually a final survival strategy which will result in cell death. Then when there is an increased ligand, there's an increased growth factor signaling on the cell membrane, then the cell will take up a lot of nutrients such as glucose and amino acids. These nutrients will be allocated into metabolic pathways. Then you get a lot of energy production and also a lot of synthesis of macromolecules such as proteins, lipids, and nucleotides. The end result is then an increase in biomass, just actually cell proliferation and the formation of daughter cells, the sear cell growth and the formation of daughter cells with the cell proliferation. Here you can see the difference between the metabolism of a cell at rest and the metabolism of a proliferating cell. When the cell is at rest, there is actually a basal rate of glycolysis. The glycolysis is the conversion of glucose to pyruvate. Then the pyruvate is taken up by the TCA cycle, and there it is oxidized by oxidative phosphorylation. It's actually broken down. These cells can also take up other substrates such as free fatty acids and amino acids, This can also be oxidized by oxidative phosphorylation. Thus, in a cell at rest, the majority of ATP of energy comes from oxidative phosphorylation. Then, in a proliferating cell, thus also in a cancer cell, there is a large increase in the glycolytic rate. The glucose is taken up and converted to pyruvate. Here, most of the energy is generated. Then, most of the pyruvate is converted to lactate, which then is secreted from the cell. And this reaction from pyruvate to lactate regenerates actually the energy which is necessary to maintain the high glycolytic rate. Then also slowly the pyruvate is taken up via the PCA cycle. The pyruvate is converted to acetyl-coenzyme A, which is then converted to citrate. And citrate is actually necessary for the synthesis of lipids, which are necessary for the formation for the lipid membranes of the daughter cells. Then what is now the big difference between a cancer cell and a normal cell? Well, a cancer cell consumes glucose at a very high rate, there's a high glycolytic rate, and it secretes most of the glucose-derived carbon as lactate, rather than oxidizing it completely via oxidative phosphorylation. What is now the advantage for a cancer cell of the high glycolysis? Well, the percentage of energy is higher than that produced from oxidative phosphorylation because you have such a high glycolytic rate, and also the breakdown of glucose delivers intermediates which are necessary for the biosynthesis of macromolecules such as lipids and proteins. This figure actually shows the signaling network which leads to the metabolic phenotype of the cancer cell. Thus you actually have an increased activation of the PE3K ACT-mTOR pathway. Thus when the growth factor binds to its receptor, PE3K is activated, which then activates ACT and mTOR. This leads to an increased uptake of glucose in the cell and to an increased uptake of essential amino acids, which then again induce an increased protein translation. Then the increased activation of this pathway also leads to an increased translation of HIF-1 alpha. In normal circumstances, HIF-1 alpha is degraded, but under conditions of hypoxia, which finds place when you have a tumor environment, then HIF-1 alpha is not broken down. Then the activation of HIF-1 alpha leads to the activation of different genes, namely to an increased expression of glycolytic enzymes, an increased expression of LDH1, which leads to the conversion of pyruvate to lactate, and it also leads to the expression of PDK1, an enzyme which is responsible for the conversion, which inhibits the conversion of pyruvate to acetyl-coenzyme A. Then the transcription factor MYC, it also induces an increased expression of glycolytic enzymes, an increased expression of LDHA, and an increased expression of enzymes which are necessary for the synthesis of nucleotides. Thus, overall, the signaling network in the cancer cells leads to an increased glycolysis and increased production of macromolecules. Then, actually, what is the role of glutamine? Well, this is an amino acid which matches the influx of intermediates to maintain the biosynthesis of macromolecules. Thus, like I just said, citrate is necessary to form lipid membranes for daughter cells. Thus here we have a net loss of oxaloacetate. Here the metabolism of glutamine plays an important role. Glutamine can give nitrogen atoms to purines and pyrimidines in the synthesis of nucleotides. Then glutamate is formed. Glutamate can then give its amino groups to alpha keto acids, which then form amino acids and alpha-ketoglutarate. Alpha-ketoglutarate can enter the mitochondria, where it is further oxidized by the PCA cycle to oxaloacetate, and then again citrate can be formed from oxaloacetate to form the lipid membranes of daughter cells. Actually, the cancer metabolism is a little bit of uncharted territory. There are a lot of questions that still need to be answered. One of these questions is, do cancer cells use cancer-specific pathways, metabolic pathways, or combinations of pathways? with maybe pathways that are different from proliferating cells. And two cancer cells require different stages of metabolic adaptations to progress from the primary tumor stage to invasion and metastasis. There's actually, is there a difference in the metabolic phenotype from stage 1 lung tumor and a stage 4 lung tumor? The answers to these questions can lead to important diagnostic and therapeutic possibilities. It can lead to new detection methods because metabolite derivatives can be used for molecular imaging of cancer. It can lead to a new diagnosis technique because we can develop new diagnostic tests, and it can improve the therapy for lung cancer maybe because maybe we identify new drug targets. Then I come to the hypothesis of my research project. We think that the problem is very clear right now, namely that there is an urgent need for effective tools that enable an early detection of lung cancer. We actually think that analysis of the metabolic phenotype by NMR spectroscopy will permit the early detection of lung cancer. What are we going to do to prove this hypothesis? Well, we will collect fasting blood samples from lung cancer patients and from control patients. Then we analyze the metabolic phenotype of these blood samples by means of proton NMR. Then by means of statistical analysis, we are going to investigate whether there is a discrimination between the metabolic phenotype of lung cancer patients shown in red and of control patients shown in black. Then, of course, we need to identify which targets and which chemical pathways are responsible for this discrimination. Then I come to my first results of the study. This slide actually shows the subject characteristics of my first group. We collected already blood samples from 79 lung cancer patients and from 78 patients in the control group. The diagnosis of lung cancer was confirmed by biopsy, of course. We actually tried to make the groups as compared as possible. Thus, in the male group, we have 67% males and 33% females. In the control group, we have 58% males and 43% females. We also tried to match for age. The average age in the lung cancer group was 68 with 64, compared to 64 in the control group. Then in the lung cancer group, we had more active cigarette smokers than in the control group, and of course more never smokers in the control group than in the lung cancer group. Then this slide shows that we collected blood samples from 58 patients with non-small cell lung cancer. 29 patients had an adenocarcinoma, 22 spinocerebellar carcinoma, 3 patients had a large cell carcinoma, 3 patients had adenosquamous carcinoma, and 1 had a carcinoma tumor. And also 14 patients had a small cell lung cancer, and 7 patients had lung cancer with an unknown histological diagnosis. Then the stages, we collected blood samples from 79 lung cancer patients. 22 patients had a stage 1 lung cancer, 9 patients had a stage 2 lung cancer, 25 patients had a stage 3 lung cancer, and 23 patients had a stage 4 lung cancer. Then when we compared the proton NMR spectrum from a lung cancer patient and a control subject, actually by visual comparison, we can conclude that we actually cannot see any systematic differences between these two spectra. But when we do statistical analysis, we can conclude that 31 of the 96 variables that we have identified are significantly different between the blood plasma of a lung cancer patient and the blood plasma of a control subject. We can see that most of the metabolites are significantly upregulated in the blood plasma from lung cancer patients, shown in black, but also some metabolites are downregulated in the blood plasma from the lung cancer patients. Then when we did multivariate statistical analysis with the results of the complete NMR spectrum, just with all the 96 variables, we can conclude that we actually can see a very good discrimination between the lung cancer group shown in red and the control group shown in black. We can actually conclude that these two groups can be can be discriminated with a specificity of 95% and a sensitivity of 87%, that actually are very good results. Then we try to construct a statistical model which permits an optimal discrimination between the lung cancer patients and the control subjects. That we actually make a model which consists of 39 variables, and with this model we actually can get a discrimination to a specificity of 96% and a sensitivity of 94%. Then, of course, we need to evaluate the results of the constructed model in an independent study population. On this figure, you can actually see the first preliminary results of a group of 22 lung cancer patients and of 22 control subjects. We can also see a very good discrimination between the two groups. We can, based on results of the statistical model, we can discriminate between these two groups with a specificity of 100% and a sensitivity of 91%. Of course, we still have to collect a lot of blood samples to verify these results in a bigger patient group. Then in the future, of course, we still have a lot of work to do. We need to examine which metabolites and which biochemical pathways are involved in the development of lung cancer. We need to evaluate the results of the constructed model in an independent study population. Therefore, we will collect blood samples from 250 lung cancer patients and of 250 control subjects. Then, of course, we want to investigate whether the metabolic changes in the blood correlate with the metabolic changes on the PET-CT scan, and we want to investigate whether the patients who do not react on a specific therapy, called non-responders, can be discriminated from patients who do react on a specific therapy, called responders, by means of proton NMR. Well, this was my presentation. Thank you for your attention. Are there any questions?